Is screen visible?
Yes, sir.
Okay. Now today we will discuss about RNA editing. Uh, जैसे कि मैंने अल्टरनेटिव स्लाइसिंग के केसेस में बताया था कि पर्टिकुलर एक सिंगल एमआरएनए ट्रांसक्रिप्ट मल्टीपल प्रोटीन प्रोडक्ट्स फॉर्म कर सकता है. Similarly, in the case of RNA editing, it is means RNA editing is another way of altering the sequences of mRNA. RNA editing is also known as RNA modification, and it is a molecular process through which some cells can make discrete changes to specific nucleotide sequences within an RNA molecule after it has been generated by RNA polymerase, means after the process of transcription. Generally, RNA editing is occurs in all living organisms, and it is one of the most evolutionary conserved properties of RNA. मीन्स आरएनए एडिटिंग नियरली सभी ऑर्गेनिजम्स में होती है नेचुरल प्रोसेस के थ्रू, क्योंकि जनरली पर्टिकुलर मोस्ट इवोल्यूशनरी कंजर्व प्रॉपर्टीज है आरएनए की, तो जनरली ये पर्टिकुलर एडिटिंग प्रोसेसेस चलती रहती है. मीन्स आफ्टर द ट्रांसक्रिप्शंस एमआरएनए जो फॉर्म होता है वो पर्टिकुलर एडिट होता है. RNA editing, just like a splicing, can change the sequence of an RNA after it has been transcribed. Thus, the protein produced upon translation is different from that predicted from the gene sequence. Now, there are two mechanisms that mediate this RNA editing process. First one is the site-specific deamination, and second one is the guide RNA-directed uridine insertion or deletion. First one is the site-specific deaminations. जैसे कि नाम है इसका साइट स्पेसिफिक डीएमिनेशन, मींस एक किसी पर्टिकुलर स्पेसिफिक साइट्स पे पर्टिकुलर डीएमिनेशन की प्रोसेस होगी. इन दिस केस यहाँ पे पर्टिकुलर जो आपका साइटोसिन, जो एग्जांपल अभी आपको बताएंगे मैमेलियन एपोलाइपोप्रोटीन बी जीन का, जनरली इस केस में क्या है, साइटोसिन का डीएमिनेशन होता है, और साइटोसिन के डीएमिनेशन से क्या फॉर्म होता है, जनरली यूरेसिल. साइटोसिन का जब डीएमिनेशन होता है तो क्या होता है, यूरेसिल फॉर्म होता है. डीएमिनेशन प्रोसेस में क्या होता है, मींस रिमूवल ऑफ अमीन ग्रुप. रिमूवल ऑफ अमीन ग्रुप मींस पर्टिकुलर अमाइड ग्रुप जो आपका NH2 है वो वहां से रिमूव होता है, तो पर्टिकुलर क्या होता है, यूरेसिल फॉर्म होता है. तो साइटोसिन से अगर अमीन ग्रुप रिमूव होता है तो वो यूरेसिल में कन्वर्ट हो जाता है. तो in this case, in this site-specific deaminations, a specifically target cytosine residue within mRNA is converted into uridine by deamination process. This process occurs only in certain tissues or cell types and in a regulated manner. Means ये सभी सेल टाइप्स या पर्टिकुलर सभी सेल्स में मतलब ये प्रोसेस नहीं होती है. रेगुलेटेड मैनर में स्पेसिफिक टिश्यूज या सेल टाइप्स में ये प्रोसेस वहाँ पे गवर्न होती है. For example, mammalian apolipoprotein B gene. This apolipoprotein B gene product means apolipoprotein B play a role in the lipid metabolism. It transport the triglycerides and different dietary proteins जो है उनको ये transport करता है सेल के अंदर. तो particular this gene, the apolipoprotein gene has several exons. Approx 2156 exons present होते हैं इस particular genes में. This gene has several exons within one of which is a particular CAA codon. Means out of several exons, one codon is a CAA. Yeah, and it exon 26 has this CAA codon. Exon 26 नंबर पे जो आपका exons है उसमें particular एक CAA codon होता है, and this codon is targeted for editing. The deamination process in the site-specific deamination, and as in this example, carried out by the enzyme cytidine deaminase, which convert the cytosine to uracil. The enzymes which deaminate the which convert the cytosine to uracil after the deamination is cytidine deaminase. The deamination occurs in tissue-specific manner, as I previously said. It occurs in a tissue-specific manner. Meshes are edited in intestinal cells, but not in liver cells. Means this type of deamination occurs only in the specific manner. Means, for example, the deamination occurs only in intestinal cell, not in liver cells. Here are the examples. This is the pre-mRNA. which have approx 2153 codons, and within exon 26 there is a CAA codon. These dark color part are the exonic regions and light color part are the intronic regions. After splicings, these all the codons are all the exons are ligated and form a mature mRNA. Where in exon 26 there is a CAA codon, and this codon will be edited with the help of cytidDNase enzymes. As I said that this editing occurs in a tissue-specific manner, means there is no change in the liver cells, but editing occurs only in the intestinal cells. In the liver cells, where no editing takes place, this mRNA, this is the mRNA which translate and form a proteins, and these proteins involved in the transport of endogenously synthesized cholesterol and triglycerides. The proteins formed in liver by this mRNA involved in the transport of endogenously synthesized cholesterol and triglycerides. Whereas in the case of intestinal cells, this CA codons, in which cytosine is present, and by the action of cytidine deaminase enzymes, this cytosine is converted into the uracil, and then after the deamination process, or after the editing process, this CA is converted, which as this CA codon, which code for the amino acid glutamine. But after the deamination process, cytosine is converted into the uracil, and then this CA codon is converted into an stop codon. Here is the UAA, means C is converted into the uracil, and then CA is converted into the stop codon. And then when translation takes place, there is a truncated proteins, means starter protein is formed, and these proteins involved in the transport of dietary lipids to the various tissues. Means shorter version are formed in intestinal cells and longer version are formed in the liver cells. The longer versions involve in the transport of triglycerides and particular cholesterol and triglycerides, whereas shorter versions are involved in the transport of dietary lipids to various tissues. Means a single mRNA transcript produce two different products which involve in two different functions. The second example of this deamination process is the mRNA editing by enzymatic deamination, include adenosine deaminations. Means first one, as in the case of mammalian apolipoprotein B, where cytosine is deaminated in the form uracil. In this case, the deamination of adenosine form inosine with the help of enzyme adenosine deaminase acting on RNA. This adenosine deaminase only acting on RNA, not on the DNA. It is also known as ADAR. Short form is the ADAR, adenosine deaminase acting on RNA. And this ADAR, which there are three in humans, produce inosine. Means three types of ADAR are found in the humans, which convert the adenosine into the inosine. This inosine can base pair with the cytosine, and this change can readily alter the sequence of protein encoded by mRNA, as see previously in the case of cytosine deaminations, where protein structure is changed after the editing process. Similarly, inosine can base pair with the cytosine, and then cytosine, and this change can alter the sequence of protein coded by mRNA. Means adenosine can base pair with the normal adenosine. In the normal case, adenosine base pair with the thymine, but after deamination, adenosine converted into the inosine, and this inosine can base pair with the cytosine. Means the particular proteins after the particular protein found by this mRNA is changed. The example of this type of deamination process is an ion channel which expressed in the mammalian brains, and this ion channel is targeted for the gene coded this ion channels is a target for this type of editing. Generally, a single edit in its mRNA elicits a single amino acid change in the protein. A single edit means a particular in a particular codon where A is present and then A is converted into the cytosine by ADAR. Then a single amino acid change in the protein, which turn alters the calcium permeability of the channel. In the absence of this editing, brain development is seriously impaired. Means अगर इस ये editing नहीं होती तो particular brain properly वहाँ पे develop नहीं होगा and then further impaired situation पे रहेगा. Means ये it is a natural process where this type of RNA editing takes place. The second mechanism of RNA editing is guide RNA directed uridine insertion or deletions. A very different form of RNA editing is found in the RNA transcripts that encode proteins in the mitochondria of trypanosomes. In this case, multiple uridine residue are inserted into a specific region of mRNA after transcription. Means particular specific sites पे multiple uridine residue यहाँ पे add किए जाते हैं with the help of guide RNA. These insertions can be so extensive that in an extreme cases, they amount to as many as half of the nucleotides of the mature RNA. The addition of uridine to the message changes codon and reading frames, completely altering the meaning of the message. अगर पर्टिकुलर यू रेसिड्यू का इंसर्शन होता है तो पूरा का पूरा फ्रेम क्या हो जाएगा, शिफ्ट हो जाएगा एंड देन फॉर द फ्रेम शिफ्ट होगा वहां पे तो पर्टिकुलर जो एक्चुअल प्रोटीन प्रोडक्ट नॉर्मल ट्रांसक्रिप्ट के साथ फॉर्म होती उसकी वजह वहां पे डिफरेंट प्रोटीन्स वहां पे फॉर्म हो जाएंगे. मींस एक्चुअल जो मीनिंग है मैसेज की वो पूरी तरह से चेंज हो जाएगी. फॉर एग्जांपल in the case of trypanosome COX2 gene, in a specific region of the mRNA of this gene, 4 uridine residue are inserted between adjacent bases at 3 sites. Means in this case, trypanosome COX2 genes, 4 uridine adjacent bases at 3 sites, 2 uridine at one site and 1 U at each 2 additional sites. Means total 4 uridine residue are inserted with the help of guide RNA directed uridine insertion method. How these additional bases are inserted? Generally, uridine are inserted into message by so-called guide RNA, gRNA. These gRNA, generally the length of these gRNA range from 40 to 80 nucleotide and encoded by the genes distinct from those that encode the mRNA they act on. Means these gRNA encoded by different genes which act on the particular mRNA which will be edited. Each guide RNA is divided into 3 regions. First one is the anchor regions, which present at the 5' end of the guide RNA, and it directs the guide RNA to the region of the mRNA it will be edited. Means it directs the guide RNA to the region of mRNA which will be edited. The second one, the second regions determines exactly where uridine residue will be inserted within the edited sequence. Means first one, जो एंकर रीजन है वो पर्टिकुलर गाइड आरएनए को पर्टिकुलर डायरेक्ट करता है कि कौन एमआरएनए के किस रीजन को एडिट करना है. सेकेंड रीजन जो है वो पर्टिकुलर जो आपका है वो पर्टिकुलर डिटरमाइन करता है कि एग्जैक्टली यूरिडीन रेसिड्यू को ऐड कहाँ पे इंसर्ट कहाँ पे करना है. थर्ड वन इज द 3' at 3' it is a poly U residue. The anchor region of guide RNA contains the sequence that can base pair with the region of the message immediately besides 3' beside the region that it will be edited. Means जो anchor region है guide RNA का वो particular, here is the, this is the anchor region and this is the editing region. Anchor region just means अगर insertion यहाँ पे होना है, जैसे इस केस में यहाँ पे अगर यहाँ A के बाद में G के बाद में और A के बाद में, तो ये particular जस्ट एडिटिंग रीजन से जस्ट पहले एंकर रीजन आपका होता है गाइड आरएनए का, जो कि पर्टिकुलर डिटरमाइन करता है कि किस रीजन मींस इस रीजन में आपका एडिटिंग होना है, तो उसके जस्ट पहले आपका पर्टिकुलर ये गाइड आरएनए का एंकर रीजन यहाँ पे होमोलॉजी शो करेगा और इस एमआरएनए से इंटरेक्ट करेगा, बेस पेयर करेगा एंड देन फर्दर इसको यहाँ पे रिक्रूट कराएगा. ये आपका एडिटिंग रीजन है जो एग्जैक्टली बताएगा कि यू को कहाँ पे ऐड होना है. तो ये हो गया. एंकर रीजन ऑफ द गाइड आरएनए कंटेन्स द सीक्वेंस दैट कैन बेस पेयर विद द रीजन ऑफ द मैसेज इमीडिएटली बिसाइड द रीजन दैट विल बी एडिटेड. This is followed by the editing instructions. A stitch of guide RNA complementary to the region in the message to be edited but containing additional A's means जिस particular region में आपका editing होना है वो second regions को define करता है. And the third region of the guide RNA is the poly-U region. The role of this poly-U region is unclear, but it proposed that they tether the guide RNA to purine sequences in the mRNA upstream the edited region. Uh, the guide RNA and mRNA form an RNA-RNA duplex with looped out single stranded region opposite where U's will be inserted. And an endonuclease recognizes and cuts the mRNA opposite these loops. Editing involves the transfer of U's into the gap in the message with the help of enzyme 3' terminal uridyl transferase TUTase. And after the addition of U's, the two half of the mRNA are ligated by the RNA ligase, and the editing is completed here. In this case, this is the general case. It is a DNA sequence, and after transcriptions, it forms primary RNA. If editing takes place at this site, as in the case, as I said that in the case of trypanosome cox2 genes, where 4 uridine residue are inserted, 2 uridine at the one site and one here and one here, means total 4 uridine residue are inserted in this case. And after editing, in here, this is the particular mRNA which formed after the editing. Previously, before editing, this mRNA produce a different protein product, but after editing, this mRNA produce a different product because addition of U change the total reading frames, and then it forms a different protein product. This is the guide RNA, which has the anchor regions, which determines here which directs the guide RNA to edit the mRNA. Editing regions which exactly decide the where uridine residue must will be added, and this is the poly-U stage. How this guide RNA edit the particular mRNA. This is the mRNA. In this case, 2 U residue, these individual 2 U residue will be added, and he at this site, in this case, 2 U residue will be added. Here, insertion of 2 U residue already takes place at a particular individual sites, and 2 U residue will be added here. This is the guide RNA, anchor regions, region of homology which base pair with this region just before the edited regions, and this is the editing region which directs the particular editing where, exactly where U residue will be added. Here in this case, guide RNA shows the complementarity with this mRNA and then base pair with this mRNA. Base pairing of this guide RNA with this mRNA excludes this A, because loop out this A because there is no nucleotides present which base pair with this A residue. And then this is loop structure recognized by an endonuclease enzymes which cut at this sites. मीन्स ए एंड जी के बीच के फास्फोडाइस्टर बॉन्ड को ये क्या करेगा, एंडोन्यूक्लियस कट करेगा, एंड देन यहाँ पे गैप क्रिएट होगा. अब नेक्स्ट स्टेप पे ट्यूटेज एंजाइम्स जो है, टर्मिनल डीऑक्सीन्यूक्लियोटाइड ट्रांसफरेज एंजाइम्स जो वो क्या करेगा, यूरिडिल को यहाँ पे ऐड करेगा, मीन्स इन द रिस्पांस ऑफ दिस एडेनिन रेसिड्यू, एंड देन इस ए के रिस्पांस में यहाँ पे यू ऐड हो गया, एंड देन यू यहाँ पे ऐड हो गया. तो ये दो यू यहाँ पे ऐड हो गए, एंड आफ्टर दैट लाइगेज आरएनए लाइगेज एंजाइम्स क्या करेगा, इस जो निक है उसको सील कर देगा, एंड देन फॉर द एडिटिंग यहाँ पे कंप्लीट हो जाएगी. तो इट इज अ guide RNA directed RNA editing where particular guide RNA which add the U residue at a particular specific sites. This RNA guide RNA further after adding this uracil residue, this guide RNA further add more U residue at 5 to 3 directions. This RNA editing is completed, and now gene transfer in animal systems. Generally, gene transfer to animal cells can be achieved via 3 routes. The most straightforward is the direct DNA transfer. Means it means the physical introduction of foreign DNA directly into the cell. The second route is termed as transfection, and this encompasses means this transfection encompasses a number of techniques, some chemical and some physical. And third is the package the DNA inside an animal viruses. Generally, viruses are evolved mechanisms to naturally infect the cells and introduce their own nucleic acid. The transfer of foreign DNA into a cell by this route, means package of the DNA inside in animal viruses, is termed as transduction. Whichever route is chosen, the result is transformation, means a change of the recipient cell genotypes caused by the acquired foreign DNA, means the transgene. There are two types of transfections, transient and stable. In transient transfection, the transfected DNA is not integrated into the host chromosomes. DNA is transferred into a recipient cell in order to obtain a temporary but high level of expression of the target gene. Means in the case of transient infections, the DNA which the transferred DNA, particular the transgene, is not integrated into the host chromosome. It just temporary and high level of expressions can take place here. In the case of stable transfections, it is also called as a permanent transfections because the particular transferred DNA is integrated into the chromosomal DNA, and the genetics of recipient cell is permanent changed. Means the DNA, the transgene is integrated in the case of stable transfections, whereas in the case of transient transfections, the transgenes is not integrated into the host genome. Now, physical methods of gene transfer. First one is the electroporations. As in the case of electroporation, means here electric field which create a pores in the cell membrane, and then these pores help in the transfer of transgene. Electroporation is the method of transformation via direct gene transfer. It involves the generation of transient nanometer-sized pores in the cell membrane by exposing cells to a brief pulse of electricity. DNA enters the cell through these pores and transported to the nucleus. In this technique, mixture containing cells and DNA is exposed to very high voltage electric pulses for very brief time periods. It results in formation of transient pores in the plasma membrane through which DNA seems to enter inside the cells and the nucleus. Just a minute. हेलो. हाँ, क्लास ले लेते थे. हाँ. नहीं नहीं. This is the electroporation method. These are the electrode. This is the anode and this is the cathode. And then when electric field is applied, this creates a pores in the cell membranes, and these pores involve in the transfer of direct transfer of transgene or DNA into the cell. And after that, when electric field is switch off, and then these pores are further resealed, and then DNA enters into the nucleus where uh, it integrates into the genomes, or some expressions takes place here. The technique has electroporation technique has high input cost because a specialized capacitor discharge machine is required that can accurately control pulse length and amplitude. Advantages of electroporation techniques is it is a highly versatile and effective for nearly all cell types and species. It is an efficient method as a majority of cells take in the target DNA molecule. It can performed at a small scale, and only a small amount of DNA is required as compared to the other methods. The disadvantages of the electroporation technique is cell damage. Cell damage is one of the limitation of these methods because irregular intensity pulses resulting in two large pores which fail to close after membrane discharge, and then further it mm, shows an harmful effect on the cell. Means if pores are not sealed, then cells may be dead. Another limitation is the non-specific transport, which may result in an ion imbalance causing improper cell function and cell death. Means if pores are not sealed, then maybe it may be chances that there is an ion imbalance situations because some ions exclude out from the cells, and then there is an ion imbalance which cause improper cell function and ultimately cell death. Second one, physical. Second method is the microinjection. It is also as a physical method of DNA transfer. In this case, a fine glass micropipette uh, is used to manually inject the transgene at microscopic or borderline microscopic level is known as microinjections. Microinjections method in recent days, microinjection method is also used in the in vitro fertilization, just like in the case of test tube baby or IVF, in vitro fertilizers, as well as test tube baby in the case where microinjections takes place. Microinjection process is used for the uh, transfer of particular in case of test tube baby or in microinjections is used for the transfer of semen into the egg. Means the gene gametes, the male gametes is transferred female gamete with the help of microinjection method in the case of test tube baby or in vitro fertilizer method. This microinjection method involves the direct mechanical introduction of DNA nucleus or cytoplasm using a glass microcapillary injection pipette. The delivery of foreign DNA is done under a powerful microscope using a glass micropipette. Uh, Cells to be microinjected are placed in a container. A holding pipette. There is a two types of pipette. One is the holding pipette and one is the injectable pipette. And this whole process are governed under a high power microscope. Holding pipette hold the cells, and then injecting pipette, which carry the DNA which to be transferred into the cell, is injected the DNA into the cell, where DNA enter into the nucleus or cytoplasm, and then if it inserted into the nucleus, then it may be incorporated into the genome. This is the whole process. This is the holding pipette, which holds the cells with the help of suction pressure. And this is the injecting pipette. Here is the DNA solutions, which will be injected into the cell nucleus or cytoplasm. This injecting pipette injects the DNA into the nucleus or cytoplasm, and then DNA transfer takes place. Next one is the particle bombardment. This particle bombardment method is also known as the particle gun method, microprojectile bombardment method, or particle acceleration method, also known as the ballistics methods. Generally, in this case, it implies high velocity microprojectiles to deliver substances into cells and tissues. Particle bombardment procedures involves coating micrometer-sized gold or tungsten particles with DNA. Means in this case, uh, these microprojectiles. These microprojectiles are generally micrometer-sized gold or tungsten particles with coated with the DNA, which to be transferred into the cells, and then accelerating these particles into the target cells or tissues using a powerful force, such as a blast of high pressure gas. Generally, helium gas are used in this case, or an electric discharge through a water droplet. The target cell is placed in the apparatus, and a stopping screen is placed between the target cell and microcarrier assembly. The passage of high pressure helium ruptures the plastic rupture disk, propelling the DNA coated micropellets into the target cells. Here is the example. This is the Zine gun barrel. Here, a plastic disc with DNA coated gold particles means gold particle, which is a micro projectiles which coated with the DNA. Here is the gold particles which is coated with the DNA. Here is the plastic disc. Gas means high when helium gas in the presence of high pressure is applied here. This plastic disc is ruptured, and then this particular DNA coated with the gold particles are forcefully particular enter into the cells directly transfer the DNA to the cells. This is the screen which stop the these particular plastic disc, and then only DNA is enter into the DNA coated with the gold particles enter into the cells, and then transfers completed. Transfer of DNA is completed. These are the physical methods of gene transfer method. Now, chemical methods of gene transfer. Calcium phosphate mediated DNA transfer. In this case, it is a very general method of the introduction of DNA into a wide range of cell types. This type of DNA transfer method is dependent on the formation of fine DNA calcium phosphate co-precipitate. Means in this case, firstly, particular a co-precipitate is formed with the help of DNA and calcium phosphate. As calcium phosphate, if calcium phosphate is ionized, then calcium ion and phosphate, calcium with positive charge ion and phosphate with negative charge is formed, and then it calcium ions bind with the DNA because DNA has a negative charge, and then they form a co-precipitate which first settles on the cells and then it is internalized. The precipitate must be found freshly at the time of transfections. And this method is suitable only for cells growing in monolayers, not those growing in suspension or clumps, in the form of clumps. The internalizations process of this co-precipitate taken up by the endocytosis process. The internalization process with the help of endocytosis, and it transport the into the nucleus because these DNA calcium co-precipitate internalized into the cells with the help of endocytosis, and where it transport into the nucleus. This is the whole process. It is the calcium chloride solution or calcium phosphate solutions. Here is the DNA in phosphate buffer, and then particular DNA calcium phosphate co-precipitate is formed with the help of these two solutions, and then it is the cells where DNA can be DNA is to be transferred. These DNA calcium phosphate co-precipitate firstly settle on the cell surface, and then because surface of the cell surface is also negatively charged, and the calcium ions which bind with the DNA is positively charged, settle on the surface of the cells, and then through the process of endocytosis, cells internalized into the this particular co-precipitate into the cells, and then it transports into the nucleus, and then transfer takes place. Advantages of these calcium phosphate precipitations method is it is a simple and an inexpensive, generate stably transfected cell lines, highly efficient, and can be applied to wide range of cell types. It can be used for stable or transient transfections. The disadvantages: it may be toxic, especially to primary cells. Some cell lines are adversely affected by the co-precipitate due to its toxicity and hence difficult to transfect. Uh, the slight change in pH, buffer salt concentration, and temperature can compromise the efficacy of this method. It is a relatively poor transfection efficiency compared to the other chemical transfection method like lipofections. Uh, it is limited by the composition and size of the precipitate. These are the advantages and disadvantages of calcium phosphate precipitation method. Next one is the DEA dextran mediated DNA transfer. DEA is a diethyl aminoethyl dextran. and it is a soluble polycationic carbohydrate which promotes interactions between DNA and endocytic machinery of the cell. In this method, the negatively charged DNA and positively charged, because DEAE is the polycationic carbohydrate, means it has a positive charge. So in this method, the negatively charged DNA and positively charged DEAE dextran form aggregates through electrostatic interaction and form a polyplex. A polyplex is the just like DNA phosphate co-precipitate. Similar to this, the DNA and DEA dextran form a polyplex. A slight excess of DEA dextran in mixture results in net positive charge in the DEA dextran DNA complex form. Means when a polyplex is formed and if DEA is in slight excess form, in slight excess concentration, then the overall or net charge is the positive, because DE charge of DEAE is positive, then overall net charge is a positive. And this positive charged DA dextran DNA complex binds with the negatively charged cells and then internalized through the process of endocytosis. The advantages of this method is also it is a simple and inexpensive, more sensitive. It can be applied to a wide range of cell types. It can be used for transient infections. It is generally not used for the stable transfections. The disadvantages, as such, in the case of calcium phosphate, it is also toxic to cells at high concentrations. Generally, transfection efficiency varies with the cell types, and it can only be used for transient transfection but not for the stable transfection. It typically produces less than 10% delivery in primary cells. So these are the advantages and disadvantages of DE8 external mediated DNA transfer. Next method of chemical gene transfer is the lipofections. Lipofections in the case where liposomes are used for the transfer of DNA or genes into the cells. The liposomes are artificial phospholipid vesicles which is used for the delivery of variety of molecules into the cells. In spite of DNA, different types of drugs are also transported into the cells with the help of liposomes. Generally, cationic or neutral lipid mixtures can spontaneously form stable complexes with DNA that interact productively with the cell's membrane, resulting in DNA uptake by endocytosis. Generally, liposomes are used to form a fusogenic particle with DNA, means liposomes are designed and fused with the DNA molecules. These phospholipid vesicles, when mixed with the cells in culture, fuse with the cell membrane and deliver DNA directly to the cytoplasm. In this liposome mediated, where liposomes are designed, because liposomes are artificial phospholipid vesicle, means just like a plasma membranes where phospholipid bilayers are present, similar to plasma membrane structure. Liposomes are also a phospholipid vesicles, means these liposomes also has a phospholipid bilayer, and then when fused with the DNA, then it internalizes the DNA into the interior parts, and then when these fused liposomes with interact with the cells, where it uh, fuse with the cell membrane and deliver directly DNA into the cytoplasm. This low toxicity transfection method, commonly known as a lipofection. With lipofection, uh, this transfer method is known as a lipofection. It is the one of the simplest to perform and simplest method to perform, and it is applicable to many cell types that are difficult to transfect by other means. This technique is suitable for transient and stable transformation. Here is the methods. These are the therapeutic genes, and these are the expression vector and so on. And these are the cationic liposomes. These cationic liposomes are fused with the DNA molecules, and then when these fused liposomes interact with the cells, it fused with the cell membranes, and then because bilayers, phospholipid bilayers are present in this liposome cell, as well as phospholipid bilayers are present in the cell membrane, then it fused with the cell membranes, and then in form of endosome it enters into the nucleus as well as in cell cytoplasm, where it can if this is used for the expression purposes, then where it in the nucleus it forms an mRNA, and then further mRNA converted into the proteins. Uh, Next one is the biological transfer of biological methods of gene transfers. In the case of animal systems, generally biological methods which used for the transfer of DNA into this animal cells is generally viral mediated. Generally, viruses has evolved to deliver nucleic acid safely into the animal cells. The enveloped and non-enveloped viruses is used for this purpose, and in the case of enveloped virus, they deliver nucleic acid by binding to a specific receptor on the cell surface, and then by either fusing directly with the plasma membrane or followed by endocytosis by fusing the endosomal membranes. In contrast, non-enveloped virus penetrate or disrupt the plasma or endosomal membranes with specific virion proteins. In each cases, the viral genome ends up being uncoated in the cytoplasm and transported to its normal replication site, which may or may not be nucleus. Both the cases, particular viral, in the case of enveloped and non-enveloped viruses, in both cases, viral genomes ends up being uncoated in the cytoplasm. Means they uh, transfer their genome into the cell cytoplasm, where it may be incorporated into the nucleus or not. The transfer of exogenous nucleic acid into animal cells is known as transduction. Means transfer of exogenous nucleic acid to animal cell with the help of viruses is known as transduction. Generally, different viruses are used for different transformation objectives. Retroviruses for stable integrations of DNA into the host cell genome, adenoviruses for short-term infections with high-level transient expressions, and herpes viruses for long-term expressions. In spite of these, there are also many viruses which is used for the transfer of DNA into the animal cells. First one is the retroviral virus-mediated gene transfer. Generally, retroviruses are RNA viruses that replicate via double-stranded DNA intermediate. The infection cycle of retroviruses begins with the interactions between viral envelope and the host cell plasma membranes, delivering the particles into the cells. Generally, the capsid of retroviruses contains two copies of the RNA genome as well as reverse transcriptase or integrase enzymes. After infections, when retrovirus infections takes place, then after infections, the RNA genome is reverse transcribed to produce a complementary DNA copy with the help of reverse transcriptase enzymes, and it integrates into the genome randomly. The integrated provirus comprises three genes: GAG, POL, and envelope genes. The GAG gene encodes a viral structural protein, POL encodes the reverse transcriptase enzymes and integrase enzymes, and envelope gene encodes viral envelope proteins. Uh, how gene transfer takes place with the retroviruses? As in the case of retroviral genome consists three genes: GAG, POL, and envelope. Beside this, there are 5' long terminal repeats and 3' long terminal repeats, and a packaging signal sequence. Suppose here is the retrovirus vector DNA construct. Means this is the transgene. Generally, retrovirus genome consists of 5' LTR, 3' LTR, and GAG, POL, and envelope genes. If retrovirus is used as a vector for transfer of DNA into the animal cells, then these retroviral genome is particularly uh, then a particular vector construct is formed, and in this case particular GAG, POL, and envelope gene is removed from here, because if GAG Paul and envelope gene is present in this vector construct, then it can infect the cells, and then transfer of DNA cannot take place. Uh, for the purpose of gene transfer, GAG Paul and envelope genes is removed from here, and then in the space of these particular GAG Paul and envelope gene, the same size of particular the same particular in the place of these three genes, the transgene as well as a selectable marker gene, here in this case neomycin register gene, and then it forms a particular retroviral vector construct. Retroviruses are generally infect the cells, so particular if it is not packaged in a capsid structures, then it cannot used as a particular vector which infect the cells. For packaging into a particular viral particles, it requires further envelope proteins as well as capsid proteins, which encodes by the envelope and GAG genes. So for packaging, it use a packaging cell lines. In this packaging cell lines, there are two regions which have mutated form of this retroviral genome. First one, in first region, there is a GAG gene, and in this case, Paul and envelope gene is mutated. Means Paul and envelope gene is not present here. And in this region, Paul and envelope gene is present, but GAG is absent. These is the engineered particular packaging cell lines where particular retroviral genomes in two regions, retroviral genomes is engineered, and in one region only GAG gene is present and Paul and envelope is absent, and in second regions Paul and envelope region is present and GAG is absent. In both these regions, this is the psi regions, which is a packaging signal sequence, but in both both two regions, these packaging signal sequences are mutated. So in these two regions, these particular genes express and form proteins, but it cannot be packaged because the whole full genome length is required for the packaging, which is not provided by these two regions. Full genome length is provided by this vector construct, and then when this vector construct is fused with and transported into the this packaging cell lines, these two regions provide the particular viral proteins which is required for the packaging, and here is the packaging signal sequence size the present, which initiate the packaging of the particular viral particles. Means here this is the particular retroviral vector construct which contains transgenes, and these two regions of packaging cell lines provide the viral proteins required for the particular packaging of this retroviral vector construct. This retroviral vector construct provides full genome length, and these two regions provide viral proteins. And then when it transcribes, is form an RNA, and these RNA particles is packaged into the whole viral particles, and then it infect the animal cells. It transfer the this transgene into the animal cells. This is the retroviral genome mediated gene transfer. Same. The packaging cell line has two separate retroviral gene region on its chromosomes. One contains the GAG gene and the other contains the Paul and envelope genes. This is the packaging cell lines. One contains the GAG gene and second one is the particular Paul and envelope genes. In each of these inserts, transcription is driven by sequence within the 5' long LTR regions. Both viruses' DNA segments lack the encapsulation sequence, means packaging signal sequence, which is required for the packaging of a retroviral genome into viral capsid. Both these regions have mutated encapsulation signal sequences, means these particular signal sequences is not capable to provide the signals which start the packaging, as well as the full genome length is also not here. The packaging cell line synthesizes viral proteins, but because there is no encapsulation sequence within either of the retroviral mRNA, empty viral capsids are produced. The viral proteins continue to be synthesized after the transfections. Means similarly, these particular regions provide the viral proteins, and it is the provide full genome length as well as packaging signal sequence, and then it forms a packaged retroviral vector RNA, which infect the cells and transfer the DNA into the cells. Next one is the adenoviruses mediated gene transfer. Adenoviruses are medium-sized non-enveloped isoheteral viruses containing linear double-stranded DNA of approximately 36 kb. Generally, of the known two genera, only the master adenovirus can infect mammalian cells. 6 subgroups of human adenovirus have been defined based on cellular agglutination assay and genomic gene GC content. The major advantage of adenoviral vectors are that they can be purified to extremely high titers, which makes them highly suited for in vivo applications, and the efficiency of gene transfer approaches 100% if the target cells bear the appropriate receptors. Uh, Generally, in the case of adenoviruses, there are 6 early transcription units, most of which are essential for viral application, and a major late transcript that encodes capsid. Means in adenoviruses, there are some early genes and some late genes. Early genes involves the early genes codes the proteins which involves in the viral application, and late genes involves the codes the protein which forms a capsid. The generally early phase proteins involves the entry of the viruses into the host cells and virus genome to the nucleus. The late genes are transcribed from the major late promoter, and it involves the formation of gene products related to production and assembly of capsid proteins. Generally, these first generation adenoviral vectors were replication deficient. Means in this case, particular early genes, just like E1A, E1B, and often the non-essential E3 genes, are removed from this adenoviral vector, and it is known as a replication deficient because these early genes are responsible for the replication process of adenoviruses. And in this replication deficient adenoviral vectors, these early genes are removed, and then it is known as an even replacement vector. Means in the place of even genes, we can add the transgenes in the vector form. Means in the vector construct. This replication deficient even replacement vectors has a maximum capacity of about 7 kb and propagated in cell lines transfected with DNA containing even genes. Means similar to RNA, retroviral vector systems, where uh, the retroviral vector construct fused with the packaging cell lines. Similar here, these vector construct, the adenoviral vector construct means even replacement vector construct is fused with the cell lines which have the even genes. Replication process of these even genes provide the particular proteins which involves in the packaging of these even replacement vector. This is the scenario where in this case even E3 and other regions which is genes regions are removed, and in the place of these regions transgene and selective marker gene inserted in the vector construct of adenoviral systems. These vector construct transfect with the adenovirus vector DNA into complementing cell lines that express the even A genes. Means it is the packaging cell lines which have the even regions. Means it provide the even particular even genes. Sorry, product of even genes which play a role in the packaging of these viral particles. These viral particles are packaged and then infected with the target cells, where it transfer the DNA into the cells. In spite of these two retroviral vector systems, there are so many other viral vector systems which play a role in the transport of genes into the transport of DNA into the cells. As such, in the case of retrovirus, lentivirus, this is the entry pathway is the receptor binding and membrane fusions. Vector genome form is the episomals. The transgene expression is the stable adenovirus. This is the next one is the adeno-associated viruses. The entry pathway is the receptor mediated endocytosis, and generally in vector genome forms vector construct is the episomal in the case of herpes viruses. Vector entry pathway is the endocytic or membrane fusions, and this is the episomal. Means these herpes virus gene transfer is the transient transfections, and in the case of adeno-associated viruses, it is medium to long term. Means long term purposes create particular. It may be used uh, for long term purposes of DNA transfer. Adenovirus associated viruses are used. So it is the gene transfer of in animal systems. Any query?
No, sir.
So it is okay for today.